All right, let's get started. It is my pleasure to introduce the first speaker today. We have Kylie Deer, who will be presenting her research involving plants, and she researched with Dr. Kamian. Kylie, before we hear what you have to say.
Thank you for the introduction. I am Kylie Deer. I'm a member of the Kamian Lab here at Chapman University, and today I'm presenting to you our project on leaf chemical profiling of two Fabiana species and their F1 hybrids. To start off with basics, plants produce diverse chemicals. Here we only see a few, but some estimate that there are up to 1 million chemical compounds present in all plant species. Plants use these compounds to interact with the environment, such as in attracting pollinators or in repelling harmful organisms. Humans have also found uses for some of these chemicals in areas such as medicine and nutrition. The Solanaceae family includes many common crops, including tomatoes and eggplants, as well as some flowering species. Right here we have the genus Fabiana, which is the focus of our project. Fabiana, there are around 30 species of Fabiana native to Western South America. They have needle-like leaves and they produce tubular flowers in the summer. Fabiana has current uses in the ornamental industry due to its flowers, and it also has traditional uses in medical treatment of diseases. In our project, we are focusing on two species of Fabiana. We have Fabiana imbricata and Fabiana viscosa. Fabiana imbricata is widely distributed along Western South America, along the Andes Mountains, whereas Fabiana viscosa is present only in a small area in Chile. In general, we don't see that two species within the same genus cross and reproduce. For example, we have tomato and potato within the genus Solanum, but we don't see them crossing and reproducing, and that's why we don't see tomatoes in the supermarket. Luckily, in our project, we were able to cross imbricata and viscosa in order to create an F1 hybrid generation in which the plants are a mix of the two species. So our goal here is to profile the diverse metabolites and chemicals present in the Fabiana parents and their F1 hybrids, and we hypothesize to see significant differences in these chemical profiles between the hybrids and the parents. So in order to do this, we collected those needle-like leaves from 3 imbricata and 3 Fabiana viscosa plants, and we refer to these as biological replicates. We also collected the leaves from 11 F1 hybrids. These leaves were ground into a fine powder and subjected to semipolar extraction with a monophasic solvent. So this solvent is a mixture of water and organic solvents. And so what do I mean by semipolar? Well, polar compounds dissolve in water, whereas nonpolar compounds will dissolve in the organic portion of the solvent. So semipolar compounds will dissolve in both the water and organic portions, meaning this solvent can extract chemicals ranging through different polarities. We then subjected these samples to liquid chromatography, mass spectrometry with a hydrophobic column. The data was collected and then analyzed. So we detected a total of 11,309 compounds in our samples, and 271 of those were confidently identified by chemical class and name. On the y-axis here, we can see the abundance of the chemical. And on the x-axis, we have the time in minutes that the chemical came out of the hydrophobic column. Hydrophobic means afraid of water, therefore polar compounds that do dissolve in water will not stick to the column very well and will elute out first, as shown in this box. These will be followed by semipolar compounds, and finally nonpolar compounds which do not dissolve in water and are therefore hydrophobic like the column will come out last. And so you can see we extracted a range of compounds ranging from polar to nonpolar. Since 11,309 compounds is a very large data set, we tried to simplify that with this principal component analysis similarity plot, which shows the data in 2 dimensions. And this plot shows the similarities and differences between the samples. So we have each species in a different color, viscosa in pink, imbricata in orange, and the F1 in gray. And we can see that the biological replicates of each species are very similar, whereas the different species are very different, and therefore they're grouped. Another way of looking at this data is with this heat map. This heat map shows the 271 identified compounds. On the left, we have the chemicals. Each line represents a different chemical. And on the bottom, we have the different groups. So here, red represents a high abundance of the chemical and blue represents a low abundance. So in this box, we can see that these chemicals, many of them representing lipids, are very highly abundant in Imbricata, but not viscosa. In this box, we have many chemicals that are representing organic compounds. They're very highly abundant in viscosa. However, both of the parents do share chemicals that are of similar or the same abundance. In the F1, we see that none of these F1 plants resemble the parents, but they appear to have a mixture of chemicals from both of the parents. The stacked bar graph shows the same data of abundance in a different way by showing distribution by chemical class. So although we can see that the F1 is different, has different abundance from the Imbricata and from the viscosa, we see that these 6 chemical classes, mainly composed of lipids or organic compounds and lipids, have the highest abundance in all 3 of them. And we expected these differences because we saw morphological differences as well. For example, we can clearly see that Imbricata is a purple flower, whereas viscosa is yellow. And in the F1, the colors of the flowers appear to be a mix of these 2. We can also see this in the sepal length. They're very short in Imbricata and very long in viscosa. And the F1 doesn't resemble either of the parents. To analyze the data of the 271 confidently identified compounds, I split the data into 3 groups. The first in which the chemicals are highly abundant in viscosa, the second in which the chemicals are highly abundant in Imbricata, and the last in which the chemicals are highly abundant in the F1 generation. So from these box plots, we can see that the abundance is distributed, here being the very highest abundance and here being the lowest in each species. This arachidonyl ethanolamide is an example of a chemical that was highly abundant in Fabiana viscosa. It is a chemical that interacts with the brain and that gives the same drug-like effect as cannabis. Positive costumelide is an example of a chemical that was highly abundant in Imbricata. It is of plant origin and it is used in drug development to carry out a range of biological activities. And 3457 tetrahydroxyflavone was an example of a chemical highly abundant in the F1 generation. It is an antioxidant and has cell death promoting activity, which helps in cancer prevention. Another interesting compound we identified was terbutrin, which is identical to a herbicide. And since herbicides kill plants, we wouldn't normally expect a plant to be producing a substance that can kill it. So first, we will have to carry out further tests to determine that this is not a chemical contaminant from the environment and that the plant is actually producing it itself. But if it is, this provides interesting avenues for further research because we can determine if the plant is producing this, for example, to ward off other parasitic plants, and then we would be able to find if it has a method so that is developed a method so that is not affected by the chemical itself. So to summarize, we observed chemical diversity in the 2 Fabiana species, Imbricata and viscosa. We confidently identified 271 of the 11,309 compounds we detected. The main chemical classes included lipids and organic compounds, and the hybrids showed high variation in the abundance of some compounds. During this time, we have also extracted RNA from the parents and the F1 hybrids and sent those out for sequencing. We recently just got the sequences back, and here I'm showing you that we have representation of the viscosa and Imbricata parents and the F1 generations in those sequencing reads, and the reads range from roughly 20,000 to 25,000 reads in each sample. So the goal with this will be to compare the RNA sequencing reads to the chemical profiles in order to potentially determine genes that may be responsible for the producing the compounds that we identified. And so far, I just talked to you about the identified compounds. However, we have a lot of unknown compounds. So that means they haven't been identified by name or chemical class. And of those compounds, there are a lot of them that are highly abundant in either of the parents or the F1 generation, but we just don't know what they are yet. We can see that the trend is the same as the heat map I showed you for the 271 identified compounds, in which there are chemicals highly abundant in each parent, and the F1 generation appears to be a mix of both of the parents. So this is also showing that these compounds right now are just identified by the time that they came out of the column and their mass. And there are some compounds that are very highly abundant in viscosa and not in bricata, and the other way around. There are also some cases in which the compound is present in only one parent, but not at all in the other. And to further, it aims to further identify these unknown compounds, we are going to run them through high performance liquid chromatography to purify large quantities of these unknown compounds, and then we will subject them to NMR spectroscopy to gain insight into their chemical structure. And with that, I'd like to thank Dr. Haga-Patamian for his mentorship and Chapman for the funding on this research, as well as our collaborators, Dr. Amancio de Souza at UCR and Joseph Cahill at the Ventura Botanical Gardens. And thank you all for
Thank you for an interesting talk. Who has the first question? Yes, Sal.
Where did you get the flowers? Because if they grow in South Africa, is there like the botanical gardens? Is that a place they grow?
Yes, so we have a few of those specific plants that were taken from South America and now are growing in the Ventura Botanical Gardens. And so the crosses between imbricata and viscosa were carried out there.
Dr. Wasaki.
Thank you. Kylie, thank you for a great presentation. This crossing between the two species, of course, that was induced in the lab. Does that happen in nature, and is it meaningful, you think?
So no, we don't really expect this to happen in nature. As I told you, we don't see two species in the same genus crossing naturally. However, we think this may have occurred because we think that maybe the rise of viscosa as a different species is due to allopatric speciation, which is just the branching off of the species due to a geographical barrier, in this case, the Andes Mountains. So we think that gene flow may have stopped, but not enough for the crossing to not occur. Although I will say, like, we had to try a bunch of different crosses to get this to happen. So it's not like we just decided to cross them and it worked. We had to keep trying.
Other questions?
Is there time for another one?
Yes, absolutely.
So when you do the analysis, the compound analysis, do you get 11,309 compounds every time? Do you always see the 271 compounds in those plants, or does it vary?
And can you clarify, please? So
The compounds you found by HSMS, are they always there when you do the analysis, or is there variation?
well, we only completed the chemical profiling of these plants once. So this is what we're seeing from that one chemical profiling. If we're to do it again, I don't know if it would vary.
Okay.
So yeah, I'm not sure.
Thank you.
I have a question about just the sheer number of unknown compounds in there, over 11,000 compounds. How would you go about identifying and classifying that many number of compounds?
So we were planning on starting with the most abundant unknown compounds because we're expecting if they're highly abundant, maybe they have more significance to the plant and its well-being. Like I said, we would try to run HPLC. And so since we know the time that each chemical elutes from the column, we would collect it at that time. And then subject it to NMI.
Okay, so it's a very slow process then,
Yeah.
to go through each one.
Yeah.
So, any last questions? All right, let's thank Kylie again. Let's go ahead and get started with our next talk today. So we have Kacie Sakamaki. She'll be talking about Kamogen HB. She has been doing research in my research group since her freshman year. A very large amount of that, as with most students, today was done online. But she did awesome with both the transition to online and then back to the lab. Kacie, it's yours.
Thank you, Dr. Leroux. I'll be presenting on analyzing interactions between calmodulin and HIV-1 matrix protein in both N and C terminals. So a little bit about HIV. HIV has affected over 40 million people worldwide, and it targets the CD4 T cell, which is a white blood cell in the immune system, and it works in the body to defend the body against infections and cancers. Left untreated, it will develop into AIDS, which is a potentially life-threatening disease. And there are still over 1 million new cases a year, which makes HIV a very prevalent problem still to this day. The HIV-1 matrix protein, or MA, plays a crucial role in the replication cycle of HIV-1. MA is located on the N-terminus of the GAG polypeptide, pictured here on the right side here. And it contains a myristoyl group, which mediates the association of GAG to the plasma membrane. This myristoyl group is essential for the budding process to occur in the replication cycle, and without the exposure of this myristoyl group, replication cannot occur. It interacts with other host cell proteins, one including calmodulin. Calmodulin is a calcium level sensor protein, and it contains 2 EF binding sites, which bind to 2 calcium in each site. When bound to calcium, calmodulin changes from a closed to an open conformation. As seen on the right-hand side here, this is the apo conformation. And when bound to 4 calcium molecules, it then opens to an open conformation. And then when bound to other proteins, it then changes conformation again. This is an example of another protein that calmodulin binds to. It interacts with many host cell proteins. This is just an example. It is not the one we're looking at specifically, but it's an example of this conformation change. So the calmodulin and the matrix protein interaction was first studied upon the discovery that calmodulin was upregulated upon HIV infection. This is a calcium-dependent interaction. We know this because calmodulin's calcium-dependent behavior, and the conformation of calmodulin and the matrix protein was found to decrease this compact structure of MA, which triggers the exposure of this myristoyl group that I mentioned before. And because of this, we hypothesized that possible hindrance of this myristoyl group may decrease viral replication. The aim of our study is to get a better understanding of the mechanism of binding between calmodulin and the HIV-1 matrix protein. Specifically, in our study, we'll investigate the full MA protein and MA's 2 domains independently, both N and C domains. Through this, we'll try to understand how calcium concentrations affect this binding, and we will do all trials in both apo and calcium solutions. On the right-hand here side, we see the calmodulin binding sites on the MA structure. These 2 little shaded parts are the cam binding sites on the MA structure, and there's 1 binding site in each domain. This W16 and W36 are the tryptophan residues that I'll explain later in the next slide. There are fluorophores that we're looking for in our technique that we're using, and there is one in each domain, which allows us to investigate each domain independently. Fluorescence spectroscopy is the technique that we will be using in our studies. It utilizes these two fluorescent tryptophan amino acids that are found in our MA molecule. And this aromatic structure allows it to be a fluorophore and emit light when excited at a certain wavelength. In our case, tryptophan, we will excite at 290 nanometers, and it emits at around 330 to 340 nanometers. There is one in each domain, as I said before, which allows us to investigate each domain independently. And we will take this fluorescent scan, emission scan in a range of 300 to 450 nanometers. So our experimental setup, we follow this titration procedure in each trial run. We first add in the buffer and take a scan of the fluorescence. This acts as our baseline, and we will then subtract it from the rest of our data. We add in first our MA solution, which contains our fluorophore. We add this in before our camodulin because we want to keep it at a specific concentration throughout the titration procedure because it does contain our tryptophan fluorophore. And then we will titrate in the camodulin solution at increasing volumes and take fluorescence emission scans after each titration. So what we are looking for. We are looking for the changes in emission peak wavelength. Specifically for our tryptophan molecule, we will be looking for this blue shift, which is a change in the peak emission to wavelength to a shorter wavelength. It will tell us that our tryptophan residues that we're looking for that are in the CAM binding site on the MA structure has moved to a more hydrophobic environment. This will tell us that a conformation change has occurred and confirms protein binding in our case. We'll also be looking for changes in intensity. A drop in emission intensity, also known as quenching, tells us that there's possible hiding of this tryptophan fluorophore, but does not necessarily confirm protein binding. So a little overview of the experiments we ran. We ran full MA and camodulin in both calcium and APO conditions. We ran N-terminal MA and camodulin in calcium APO conditions, and C-terminal MA and camodulin in both calcium and APO conditions. So starting off with our calcium condition results, we did confirm that there was protein binding in our full MA and camodulin in the presence of calcium. This is our fluorescence data right here. We plot it with wavelength versus intensity. We start off with our first initial value at this blue, and it takes a rapid decrease in intensity and drops down. Once the camodulin concentration has hit a certain point, then you see this blue shift occurring, which is the shift of peak wavelength. And we have said before that this is a conformation change that we're seeing and confirms protein binding. Here we have normalized and integrated this data, getting rid of the wavelength factor, and we see the same initial decrease in emission intensity that we've seen on the other side, and this gradual increase of emission intensity that we know is our protein binding curve. And this protein binding curve coincides with the blue shift that we're seeing on this side. And again, it confirms protein binding. Similarly, with N-terminal MA and camodulin in calcium conditions, we see similar behaviors. We see this initial high emission intensity followed by a rapid decrease in quenching of the signal, followed by a gradual increase and blue shift occurring, which confirms our protein binding. Our blue shift is occurring, which means there's a conformation change. And again, on the right-hand side, we see this drop of emission intensity and protein binding curve, which confirms protein binding. On the other hand, for C-terminal MA and calmodulin in the presence of calcium, we do not see the same blue shift occurring. There's no peak wavelength shift, and there's no gradual increase after this rapid decrease in intensity. This tells us that there's no conformation change occurring and no significant protein binding occurring in the presence of calcium. Moving on to our APO condition results. We see very similar results in all of these trial runs because of the calcium dependency of this interaction. Here we see the full MA and calmodulin in APO conditions. There is no presence of a blue shift occurring. We still see this rapid decrease of emission intensity, but we do not see that protein binding curve and gradual increase, which tells us that there's no significant protein binding. And again, with N-terminal MA and C-terminal MA, in APO conditions, we see the same initial rapid decrease in intensity, but no protein binding curve. So there's no protein binding. So a little analysis. When we compared all of the trials in both APO and calcium conditions, we see the same initial decrease, very rapid decrease of emission intensity. At first, our lab thought it was a mistake. We thought it was inconsistent starting values, but because of the consistency over the years, over all trial runs, we then hypothesized that it's a possible first step of a two-step binding process, or an interaction that is non-specific happens in either APO or calcium conditions. But further testing will be needed to prove this or confirm this finding. So a little overview of our findings. We have once again confirmed the calcium dependency of the interaction. We know this because of calmodulin's calcium-dependent behavior. And our results do suggest that N-terminal MA binding is a prerequisite for C-terminal MA binding. We know this because in calcium conditions, we do see the only binding of the N-terminal MA and calmodulin, and not the C-terminal MA and calmodulin. And finally, this possible two-step binding behavior that I discussed on the previous slide, but further testing will be needed to confirm this. And just a special thank you to Dr. LaRue for being such an amazing PI over these past 4 years. Dr. Owens for all the guidance and allowing us to use your lab whenever we needed it. And of course, the other Cat Lab researchers that made a huge impact on my education and for their work on this project as well. And CUE for funding our research. Thank you.
Who has questions? Yes.
I was curious, you mentioned that the mirror, is it a mirror protein or what is that?
The myristoyl group? Okay, the myristoyl group is the portion on the MA protein that binds to the plasma membrane. And I mentioned that the MA protein is part of the GAG polypeptide that is a huge structural protein in HIV-1 virus. And it has huge roles in the budding process and assembly process. And without this myristoyl group being exposed on the MA, which happens during calmodulin and MA interaction, then the MA protein cannot bind to the plasma membrane, and then budding will not begin. I can take it back if I go all the way back on this. Sorry. Right here, this is the MA protein, and then it's pictured here as this black squiggly line. But as you can see right here, then that myristoyl group binds to the plasma membrane, and without that association, then there will be no budding and essentially no replication.
Yes.
You're looking at protein-protein interactions, right?
Mm-hmm.
So can you tell me some of the advantages of using this method compared to the old-fashioned yeast to hybrid method?
Old-fashioned.
Yeast to hybrid, where you look at the interaction between two proteins. So what kind of advantages this method has?
I'm not too sure about that because I don't really know much about the other method of studying it. So I'm sorry, I cannot.
Sure, okay, let me rephrase it. Based on what you have heard by looking at this method, can you name some of the advantages of this method of looking at protein-protein interaction? What do you think is like important or useful using this method, for example?
Okay, I mean, I don't know the other method, but from this method, it allows us to see like these proteins that are so small, and I'm not too sure. I mean,
Okay, even if you're not sure, guess, that's okay. Educated guess.
just you can really tell like certain things on the protein, say like the tryptophan, like how it's moving in the environment, dependent on like the changes of emission intensity and the wavelength shifts. And I'm guessing that I only know tryptophan specifically, but other amino acids have different behaviors in this system. So if you like maybe tag it or something with a different like fluorophore, then maybe you can find out different things versus this tryptophan having this behavior shift or.
Okay, thank you.
So,
Yes.
so you talk about wavelength and intensity, and there's a detector in your setup. Is that like detecting photons that bounce off the proteins, or?
Yeah, so it emits light at a certain wavelength, and you are scanning the fluorescence at a range of, I think ours is 300 to 450 nanometers. So it's like light. It's like light at a range of wavelength.
Okay, is that from a laser?
I'm not sure. No, I don't think so. No.
Thank you.
Other questions? I have one question. We're planning on doing basically the same experiments, but this time looking at the calmodulin halves as opposed to the HIV halves. Based off these results, are there any predictions you have when we look at the calmodulin halves?
Any predictions?
In terms of binding, like so maybe certain domains won't bind.
I haven't looked at the calmodulin specifically or studied the two halves independently. Well, if one does bind over the other domain, like how we've seen in this MA, then we would know that like one domain would be like a prerequisite for the other binding site, but I wouldn't know the differences between the two calmodulins as of right now, or the two domains.
Yeah, last question.
So when the sample or the protein, it binds on its, place and it emits radiation at a, increased wavelength, how actually does that like work? I mean, if it shows you something, I think you're expecting like 300 nanometers, 400, what if it was like 600 nanometers? Like, what does it mean?
Well, so our tryptophan residue is very specific, and when we shine light at it, excite it at this wavelength, then it will emit at this wavelength. So we shine it at 290 nanometers, excite it, and it emits at 330 to 340 nanometers. And it's very specific to our tryptophan residue, and we have adjusted like our range that we're taking the data from. We've adjusted the nanometers that we're exciting it at. We have adjusted like everything to match, like get the data that we want it to be.
Okay, so thanks.
Mm-hmm.
All right, let's thank Kacie again for wonderful talk. Before we move on to Abby's talking, yeah, still a couple minutes. Wanted to just give a shout out to Davis and Kacie. Davis will be talking after Abby because they are at a much bigger disadvantage than most research students are, in the sense that they're doing this project with me. I'm not a biochemist, I'm a physical chemist. And this project was initially developed by 2 students many years ago who came to me, they're biochemistry students, and they said, we want to do a research project with you. And I said, my research is in biochemistry, but if you come up with a project, I'll support you with this project. And so they came up with both Kacie's and Davis's through a 3-month literature review. So that's how the project started. And then this project has been entirely student funded. So the students apply for funds through CEV, there's Solidarity Creative Grants, the SURF program, all that kind of stuff. So they're self-sustaining the project themselves, which is pretty awesome. So they're very good job. All right, so we'll move on to our next presenter, Aviv Bechtel. She'll be presenting on some optical metrology. This is a research project that is a joint collaboration between myself, Dr. Dressel, and Dr. Howell, who is a professor who will be coming to Chapman next year in the fall. And just like Kacie and also like Davis, she's been doing research for quite a few years in the group. Abby?
Yeah, thank you all for being here. I'm excited to be sharing with you about this research that I've done over the last 4 years. We're going to be talking about enhancing optical metrology through the use or through the manipulation of the directional asymmetry of polarizers. Before we jump in, we need to understand what optical metrology is. Optical metrology is basically the process of taking measurements using light. And it's really critical in many fields of engineering and science. And so we can take measurements of the light itself, or we can take measurements using the light, measurements of distance, angle, power, spectral properties. There's many applications of this. One photo on the left is of an optical clock. So it's very critical, lots of applications within optical metrology. But the motivation for our project comes within some challenges in this field. There's a challenge of affordability and precision with small measurements. So our goal for this project was to create an affordable, precise measurement technique using previously considered sub-laboratory quality polarizers. Because what happens is when you want to take extremely precise measurements using light, the cost of that skyrockets. So a little bit of background before I jump into our project, we have to understand what polarization is. Polarization applies to transverse waves, which are waves that oscillate perpendicularly to their direction of propagation. And light is coupled oscillating electric and magnetic fields. So we have that picture to the bottom right where we have the electric field in red and the magnetic field in blue. And so polarization is the red component of that. There are different types of polarization as well. There's linear, circular, elliptical, there's unpolarized light. And so for the purpose of this project, we're using linearly polarized light, which is frequently used in laboratory settings. And the basis of that is horizontal and vertically polarized light. And those are 2 perpendicular states. To understand a little bit more, we'll talk about the background of polarizers and Jones calculus. A polarizer is nothing more than an optical filter that allows only a specific polarization to pass through. So in the photo to the bottom right, you have all these different polarizations happening, and then you have this filter, and it's only allowing one select linear polarization through. And so we make use of this in our project. Polarization can be represented as a 2-dimensional complex vector. So this is how we model our project mathematically. We can represent the state of the light in a vector composed in horizontal and vertical components. And each optical element in our setup is going to act on that. So each element, so a polarizer, has a 2x2 matrix that acts on that light. And that's how we can mathematically model what's happening. So for an imperfect polarizer, which is the key to our project and why we can make these really small measurements, has that 2x2 matrix shown to the bottom left. So it's a normal polarizer matrix, but it has some leakage defined by eta. So it's not quite perfect. Wave plates Wave plates are another element that we use in our setup, another way to modify the light. And wave plates cause a transformation on light with a phase shift. Usually they're made of some like crystalline material with a specific bifringement. And so half wave plates, there are two general types. There's a half wave plate and a quarter wave plate. A half wave plate shifts the wave of light by the wavelength over 2, and a quarter wave plate shifts it by wavelength over 4. And so this picture here shows some light coming in at a diagonal angle in red, composed in horizontal and vertical in the blue and green, and it goes through this material, and when it comes out, it is shifted at a different angle. And so that would be an example of a half wave plate. So half wave plates change the type of light from linear to linear at some different degree, or quarter wave plates change it to then circular. And so I have the matrix elements shown for that to the bottom right. One last piece of background is understanding interferometry. So this is the setup that we're going to use, or a very simple version of it. Interferometry is a method of measurement using the interference of waves. So what we do is we take a source of light and we split it in two, and we send those two different waves of light around this box-like setup. And so we can measure the characteristics of the waves or materials that it interacts with by having these two different directions and the light interfering with each other in the two different directions. So there's a clockwise path and a counterclockwise path, and those two directions interfere. And so this is used in things like GPS, and it's been considered for a method of detecting gravitational waves, lots of applications. Oh, it is not showing up here. Or here it is, yeah, okay. So our experimental diagram, a simple version of it is shown here. We have a laser, it's coming in, we split it in half, like we saw in the previous diagram, and it comes around and it hits a mirror if we follow the clockwise direction, and it comes into this little black box with a half wave plate that we just talked about. So it'll change the linear polarization of light, a polarizing beam splitter, so it selects off a specific polarization. So we send horizontal this way and vertical gets reflected. So we select the vertical light, and another half wave plate. And we rotate this whole little square box here by one specific angle. So we rotate this by a positive angle and this by a negative of that same angle. So we can essentially consider this box as one polarizer rotated at a specific degree with a leakage eta. Then we have the same thing on the left, but in the middle is the key component of this, which is a leaky polarizer, which is really interesting because most lab setups you'd think, I don't want to use an imperfect polarizer, I need it to be laboratory quality. But the introduction of a leakage in our setup creates an asymmetry in the path directions. So clockwise is now different than counterclockwise. And so what that does is when we continue to follow the path and it goes out and then we split it into horizontal and vertical, the difference between those two becomes much closer. The difference between horizontal and vertical normally is 90 degrees apart. But if we change this angle by a small fraction of a degree, we can see that change become much closer than 90 degrees. Here is another diagram modeled using a program called Optics 3D. So it looks much more similar to what you would see in the laboratory setting. Same process. Here I show a little bit more of what we actually do. There's a telescope to collimate our beam, and then it goes through again, same process. Again, with this being the key component because it introduces an asymmetry. Some methods about our project, we started by virtually prototyping it. So together with Dr. Justin Gressel and Gabriela Nutt, we wrote code in Python to represent what we would expect experimentally. So here is a plot of the horizontal and vertical output coming from that different signal, coming from what we measure, the horizontal and vertical at the end of our setup. So then here we take a split detected signal. So we take the difference between the horizontal and vertical and over the sum of the horizontal and vertical output. And what we see is this really large peak over a super small degree range. So as we rotate that middle polarizer theta, we're going from 0 to 180, we see this really strong peak in the middle there. And that's what we want to see. With our code, we also determined the ideal angles for our two-corner, the box setups, those the halfway plates, polarizing beam splitter halfway plates. So we determined the best angles for that by iterating through all the possible combinations. So 180 degrees for both of those. And we determined that 34 degrees and 59 degrees, we see the sharpest response in that degree range. For our data that we've collected in the lab, we're in the process of doing it right now, but we've tested for angles 34 and 59. There's been a little bit of a challenge because we've gone through a couple iterations of lasers. This is a very small measurement process, and we've had some unstable lasers, so that's been a challenge. And then there's a significant run-to-run variation happening. And so as we collect samples, they've been drastically different from each other. But here's one example where we see our horizontal and we see our vertical, and we see a similar structure with this different signal here, with this sharp peak to what we saw in our computationally simulated results. It's not as strong, but it does show the same structure. For future work, obviously we want to collect further samples and just ensure reproducibility. We want to determine and minimize sources of error and quantify tolerance to imperfections in our setup, from the optical elements, from the laser itself, and then optimize sensitivity to angles, and consider alternative designs to our experimental setup, see if there are better ways that we can design. So in conclusion, what this project does is it introduces imperfect polarizers into an optical setup. These polarizers are very inexpensive, and that causes a path asymmetry, a linking of the state of the path to the polarization. And that linking of those states causes that sensitivity when we rotate the imperfect polarizer by just a fraction of a degree. We are in the process of verifying our results that we saw computationally, and the early results are quite promising, but they are inconsistent. I want to thank Dr. LaRue for all the help with this project, growing me as an experimentalist, Dr. Justin Gressel, as he has walked me through a lot of the physics of this and optics, and Dr. John Howell for coming up with this project idea, and then everyone else on the amazing research team in the past and right now, and hopefully in the future. So thank you very much. And
Questions?
Question. When you were determining that those two angles, like 39, 59,
yeah.
You said you use an iteration. Like, could you elaborate a little bit more? Like, you use a specific model to do that?
Yeah, so what I did is in code, I took all the combination of angles, so 0 to 180 for each of them, and basically looped through and found exactly where the sharpest peak happened by just taking the derivative and finding where the slope was the greatest for all combinations. Yeah.
Yes.
I'm not in this field, so I'm just curious. So
Yeah.
this noise that you're seeing, experimental noise, which is experiment variation.
Yeah.
Do you think that's coming from the fact that the polarizers are imperfect? And is that imperfection means that every time you run the experiment, you get like different imperfection?
That's a great question. We have characterized the imperfections. We've characterized all the elements so you know exactly how they act on the light. And they do consistently act on the light in the same way.
In the same wrong way, for example?
In the same wrong way. Yes.
So then that's not the reason why you're getting the noise?
No, the noise could be, because we're taking such small measurements of intensity, It could be for a number of different reasons. It could be that our setup isn't aligned perfectly. It could be variations in our laser. It could be a number of different things. Yeah.
Other questions? Yes.
Yeah.
So you said you tried out like different flavors. Can you elaborate on like what the difference is between the ones you like tried out?
Yeah, so we actually got a grant and we bought a really expensive laser that is stabilized, and so its power doesn't fluctuate and its polarization doesn't fluctuate, guaranteed up to some percentage. But that laser did fluctuate quite drastically, taking our measurements pretty difficult. Then we used some other lasers that were already in the lab, and those still had problems, so it's been a process.
One small correction. The laser was $6,000, which by scientific standards for lasers is actually very cheap. Lasers can
Cheap
go,
laser, felt expensive.
lasers can go up to the multiple hundreds of thousands.
Yes.
Um,
Compared to our other lasers.
Yes, but our other lasers are, yes.
It was expensive.
All right, one question for you. What are your future plans, if you don't mind sharing?
Yeah, my future plans, so I'm a physics and computer science double major. And after this, I will be starting full-time as a data scientist, artificial intelligence specialist at the Aerospace Corporation.
Awesome. All right, let's go ahead and get started with our last presenter. So we have Davis now presenting. He's also been in the group for slightly, I think, a month longer than Kacie, maybe 2 months longer than Kacie, so for quite a long time, over his undergraduate career. This is the other half of the biochemistry projects in my group, as I mentioned before. These were student developed, so one of Kacie was coming on, the other one that Davis was coming on. So Davis, the floor is yours.
For the introduction, Dr. LaRue, I will be presenting on exploring interactions between alpha-synuclein and chemogenic. Parkinson's disease is the second most common neurodegenerative disease. It is characterized by a loss of dopaminergic neurons, which affect voluntary movement and mood, as seen on the image on the right. Symptoms can be seen in tremors, stiffness, slowness, incoordination, and balance problems. Furthermore, the disease is characterized by the presence of Lewy bodies, which are also found in Alzheimer's and dementia patients. These diseases affect 3.4, 6, and 1 million total in the US respectively. There are no permanent cures, only symptom management, such as MAOB inhibitors, which inhibit the breakdown of dopamine, L-DOPA, which is synthetic dopamine, and deep brain stimulation. While the disease itself is not directly fatal, there is a sharp decrease in the quality of life for patients. Upregulation of chemogulin in those with neurodegenerative diseases. Chemogulin is a calcium-sensing protein and a second messenger, meaning that it plays roles in cell signaling and signaling pathways. Furthermore, it changes its shape depending on calcium, as seen in the image on the right. When it is in calcium, it is in its active state, and when it is inactive, it has a different conformation and is quote-unquote inactive. It interacts with many chemogulin-binding proteins with a wide variety of functions and locations. For example, it has actions in smooth muscle contractions, inflammation, metabolism, and apoptosis. In the brain in particular, it is involved in neurotransmitter synthesis and release, interacts with enzymes and microtubules. Alpha-synuclein aggregates forming the core of Lewy bodies. It is most abundant in the brain and is involved at the presynapse and is shown on the top. Its function is not fully understood, but it is involved in maintaining synaptic vesicles, plays a role in the regulation of dopamine release, and the movement and structure of microtubules. It also is a key component in Lewy bodies, as seen on this image. It is the yellow figure and clumps up with a bunch of other cellular components that are not properly disposed of and form these Lewy body aggregates. Lewy bodies affect dopaminergic neuron signaling and cause neurodegeneration, and there is speculative binding of Acin's amphiphilic alpha helix to chemogulin. Aims. We hope to investigate the binding between alpha-synuclein and chemogulin to determine binding affinities, conformational changes, and the role of substrate concentrations. Furthermore, we wanted to determine the effects of calcium on this interaction, as chemogulin is a calcium-dependent protein. Furthermore, we have received conflicting results from previous studies within this lab and in literature as well on the apo-calmodulin binding to ACE. In total, we hope to improve our understanding of Lewy body formation using fluorescent spectroscopy. As Kacie introduced earlier, this phenomenon is when you use light to excite an electron from the ground state to an excited state. And after some time, as the electron cannot stay in the excited state, it falls back down to the ground state, emitting a photon of light. You can think of it like playing wall ball if you are the light particle, exciting the electron into the excited state or the wall, and the ball will bounce back to the ground state. Here's a flowchart of our methods. We use two different titrations, one in apo conditions and one in calcium, apo meaning without calcium. We fluorescently tag our ACIN protein and set it as our titrant in the sample cell, as shown on the right. We add small aliquots or a titration of the calmodulin protein, and between each addition, we take a fluorometry reading. On the right is a diagram of our fluorometer, or a general overview of our fluorometer. There's a light source that excites the sample cell and a photodetector to get the readout from the emission of our fluorophore, and that is shown on our readout. Changes in signal intensity can infer conformational changes. This is how we are able to extrapolate meaning from my data. The spectra is indicative of our fluorescent tag local environment, and decreased emissions infer tag interference, and thus protein folding. For example, if we had a fluorescent tag on the far left unfolded molecule, and we see the spectra, we expect it to be widely different from when it is in its folded position, as light would have a much harder time reaching our fluorophore. Furthermore, we would see something called quenching, which shows a decrease in emission between each fluorescent spectra as we increase our titrate. Results. On the left is our apo condition titration, and on the right is our calcium condition titration. We see a much larger quenching in our calcium conditions, and the runs are color-coded at the same concentrations of our titrant calmodulin. Overall, we see a much larger quenching in calcium conditions, which is consistent with previous data, and the data strongly supports alpha-synuclein and calmodulin binding. We wanted to extract our binding affinity analysis from our fluorometry data and created a protein binding curve. On the y-axis is the fraction bound, with 0 being 0 ACIN bound to calmodulin, and 1 being fully saturated, meaning all the ACIN has bound to calmodulin. Run 1, 2, and 3 were all in calcium conditions and are shown in gray, blue, and yellow respectively. The gray and blue runs are very consistent with each other, while our third one was a little slightly away from those other two and slightly skewed our average. And the apo titration is shown in pink. We wanted to extract the KD, which is a value at which half the protein is bound. So if you intersect the 0.5 value on the y-axis with each run, you get our KD. And lower KDs indicate higher binding, as there is less concentration of substrate needed to reach half max binding. So our important values are our average calcium condition KD at 0.15 micromole per microliter, and our apo condition was at 0.523 micromole per microliter. Comparison of apo titrations. As I earlier explained, we have conflicting results from our lab as well as in literature. So I wanted to compare it with our previous researchers. My data on the far right is most consistent with Jolie's data in 2018, indicating there was minimal interactions with apo-calmodulin and alpha-synuclein. However, Napo's data in 2019 showed larger quenching, and recent data collected in our lab shows that this might have been due to protein denaturation or contamination in our buffers. Conclusions. We have successfully determined the binding affinity between ACIN and calmodulin with our average calcium KD at 0.145 micromole per microliter, and in apo conditions, a KD of 0.523 micromole per microliter. Therefore, our calcium conditioned calmodulin bounded about 3.6 times stronger to alpha-synuclein. Future work, we hope to increase our consistency of our calcium data, as that yellow line skewed the app quite far, and gather further apo titration data, as we were only able to successfully do one run. Furthermore, we'd like to explore the individual halves of chemologically to see which parts bind strongest to alpha-synuclein. Applications of our findings. We suggest that researchers use CAM's active form for crystallography when bound to alpha-synuclein and to target alpha-synuclein binding for targeting drugs when it is bound to the active state compared to the inactive state as it bound strong. Acknowledgements. I'd like to thank my PI, Dr. LaRue, Dr. Cedric Owens, and previous lab members, Justin Nako and Riley Kendall, current lab members, Kacie Sakamaki, Andrea Sandoval, and Julie Tran, and our funding support from the Chapman CUE and the NIH and IDDK. Thank you for listening. Please feel free to ask questions or reach out with any questions.
What questions do we have?
Thank you, Davis. That was really interesting. I think I have two questions. How well are you able to control the level of calcium in the system? When you say that there is no calcium added, is that like a 0% or is there always some? Because calcium is everywhere.
Yes, so we don't take our samples from cells. So we basically buy our proteins in buffers that do not contain calcium. And when we make our own buffers to run it in, we can control how much calcium, of course, we put into there. So since we're not taking it from a human or a cellular organism, there's no excess calcium that could be.
Okay, that makes sense. My second question is, I understand that these molecules are very small. Correct me if I'm wrong. Are you looking at the entire? Is the size of the two interaction partners, is it the entire size or are you looking at fragments?
I believe our chemogulin protein is 1 through 178 residues, and our alpha-synuclein is about 29 residues, including our fluorescent tag at the end. So it's not the full alpha-synuclein protein.
Would you then, is that important? Would you expect a difference if you were, could you do this with the entire protein? Would it be different?
We could definitely find that in the data. However, we're inferring that this small portion of the protein indicates larger tendencies of the full protein.
Thank you.
Yes.
I was curious, you mentioned that like, your data was possibly different from the literature. Do you know maybe why that could be?
Sorry, could you repeat the question?
Oh, yeah. You mentioned like a couple slides back about how this was different than what you would have expected. Do you know why that might be?
So the literature has two parts. Some say that there is strong binding in APO conditions, and some they say that there is not strong binding in APO conditions. So I guess either way, the data would correspond to some of the literature, but not all of it. And we wanted to further characterize which one is true because they can't both be true. They can't be strong binding and no binding. So therefore, my data strongly correlates that there is weak or very little binding, while Justin's data, or Nako's data in 2019 shows a much stronger binding. So that way, these two don't necessarily agree with each other.
Another question? I have a question about the calcium data, the run. Do you have any indication as to why run 3 was so much different than runs 1 and 2? Because runs 1 and 2 pretty much overlap perfectly. Like it's very reproducible, very nice data, but then run 3 is quite a bit shifted down.
So that could be due to residue left in the cuvette. As I remember, I took all 3 of those measurements on the same day, and run 3 would have been the final and last experiment using the same cuvette. So maybe there could have been issues with washing it. Also, the timing of each titration is a little messy, as the spectra could also be dependent on how much time you let the solution stir and mix and things like that.
You have the last question? Kacie.
Do you hypothesize that like strong binding or weak binding and why? Like what's actually going on? Which one is true based on like homogeneous behavior? Because
So I speculate that there is strong binding in calcium conditions and relatively weak binding in APO conditions.
And last question, when I'm sharing this, what are your future plans?
So I will be going to the University of Wisconsin-Madison for a PhD in comparative biomedical sciences.
All right, welcome to the second session of talks. We have 4 great speakers today, and starting with Jillian Fahey, who is doing research for Dr. Safi, or has done research for Dr. Safi, and she'll be talking about how exosomes from breast tumors induce field cancerization of non-cancerous cells in the breast. Thank you.
Thank you. So I'm going to be talking about breast cancer and then the recurrence of secondary tumors after the removal of the tumor in the breast. So why should we care about breast cancer? About 1 in 8 women will be diagnosed with breast cancer in their lifetime, and this is the most common cancer diagnosis in women. As of right now, there are only 2 current surgical treatment options, one of which is invasive, which is a full mastectomy, which is removal of all the breast tissue. This is, like I said, more invasive and leads for a longer recovery period. The second is a lumpectomy, which is what we're going to be focusing on today because it's less invasive. It only removes the tumor in the part of the breast. So an example in this image, it would remove just this tumor instead of the entire breast. And this is an advantage because it leads for a less recovery period and it allows women to still maintain their breast tissue. So in this case, though, we're discovering how exosomes might play a role in this, in the field cancerization of these adjacent tissues. That leads to what is breast field cancerization. It's the genetic, epigenetic, and biochemical alteration in structurally normal cells and tissues adjacent to breast tumors. So that means you remove the tumor and then nearby tissues adjacent to where that tumor was are now cancerous. And what we believe is causing this is the role of exosomes. And exosomes are thought to play a role in breast field cancerization as they can travel from a cancerous cell to a non-cancerous cell. Additionally, cancerous tumors are known to secrete more exosomes than non-cancerous cells. And so what we're thinking is that these exosomes, which are in the cancerous tumor, are causing this induced field, this field cancerization to produce or induce cancer within those normal healthy tissues. And this is because exosomes are released from cells containing RNA, DNA, and proteins. They are taken up by adjacent or distant cells and can affect cell function and behavior. So in this image here, this would be an example of a cell. And in our case, we'd say that this is a cancerous cell releasing cancerous exosomes. And then back here are 2 different, in our case, healthy cells. Then they could take up these cancerous exosomes and therefore causing them to become cancerous themselves. The objective of this is to better understand the mechanism of secondary tumors for enhanced prognosis and treatment of breast cancer in the future. This image here is showing the case of a tumor and then these adjacent tissues. And then here you have a lumpectomy, which is removal of just the tumor. And then you have a reoccurrence of tumors popping up sometime after. And our thought is that these exosomes are playing a role in this because if you remove just the tumor, a lot of women are still having reoccurrences. And so the exosomes might be left behind and they are in fact causing or possibly causing this induced field to promote cancerization in those adjacent tissues. So we hypothesized that the exosomes produced by breast tumors induce field cancerization in their normal adjacent breast tissue. And this was tested in human breast epithelial cell models. These cell models, there are 3 that we use. The first is MCF7, and these are cancer cells. These reflect early stage disease because they express the estrogen receptor, the progesterone receptor, and HER2, meaning that they're less aggressive because they can respond to drugs targeted directly to these receptors. MCF7 is what we use for treating our non-cancerous cells because it's more relative to what we would be exploring in adjacent tissues. On the other hand, we have MDA cancer cells. And these do not express any of those receptors, meaning that they're triple negative, which makes them more aggressive as they can be resistant to these drugs. And this is a metastatic late stage form of cancer. And then lastly, we use MCF10A healthy cells. These are non-cancerous, and this is what we were exploring to see as our adjacent tissue example. So our approach, we started by culturing the MCF10A, MDA, and MCF7 cells. And then we used their supernatants and did ultracentrifugation to isolate the exosomes from the supernatants. Once this was done, we wanted to characterize the exosomes and make sure that we actually had exosomes present. So this was done using dynamic light scattering to confirm the exosome size. So this is the dynamic light scattering showing the MCF10A, these are the healthy cells, and then MCF7, these are the cancerous cells, and then their diameter. As you can see, we have a mean of about 169 nanometers for the healthy cells and then 182 for the cancerous cells. Both of these ranges are what we would expect normally for these exosomes. And so in this case, we're really just looking at MCF7 cancerous exosomes because these are the ones that we're going to be working with. We end up just working with the cells of MCF10A, not their exosomes. But we wanted to make sure that as long as the number was within a range, that this could confirm that the exosomes were present. So once we found out that exosomes were present, we wanted to look at the proteins within them and see if they had tumorigenic qualities. So we did an SDS page size separation and then a Western blot that uses specific antibodies for detection and chemiluminescent visualization. And we were able to isolate both the exosomes of both cancerous cell lines and the cells, and then able to look at these different proteins within them. So first we have the CD63, and CD63 is just a marker that confirms that exosomes are present. And we can see that CD63 is present in both the cells and exosomes of both cancer cell lines. And this indicates that, like we saw with the DLS, exosomes were present, and we could move on knowing that we were working with the exosomes we wanted. Then we have fatty acid synthase, and the cells and exosomes also express fatty acid synthase, and this works to help with tumor growth. And so that would relate to what we were studying, and that would correlate with what we're thinking, that it expresses something that will relate to tumor growth. And then we have AR, an androgen receptor, that does about the same thing with increasing cancer cell growth. We used fatty acid synthase in AR because it's been shown in previous literature in prostate cancer that the exosomes contain both of these markers. So we isolated exosomes, and then once we isolated exosomes, we looked at their proteins and saw if they contained the ability to have tumorigenesis. Then after that, we wanted to look at their mRNA to see if that also contained the same. So we did an RNA isolation using spin column chromatography and then QRT PCR. And then here, this is showing what happened, the results for QRT PCR. Overall, we see robust expression of the fatty acid synthase, the AR, MYC1, and then actin. AR and FASN, like we saw previously in the proteins, those have the same role. And then we have 1/CT values. So basically, overall, the lower these bars means the less RNA present. So the higher bars mean that there is more mRNA present that we saw. Starting here with the actin, this is a housekeeping gene, so this is another thing that just confirms that we're looking at the exosomes, and it should be present in exosomes of both cancerous cell lines once again. And this confirmed that we could move on and that the rest of these results were accurate, looking at the exosomes. So then we have EGR1, and EGR1 results were actually inconclusive because the signal was too low. However, as you can see, it looks as though the bars are lower than the rest of these, which would mean there's less RNA present, which would correlate and make sense because it is a tumor suppressor. We wouldn't expect a tumor suppressor to be present in these cancerous cells. So even though these results were not, they were inconclusive, it would correlate with what we would expect to see. Then we have MYC1, and this also helps to increase metastasis, and we see robust expression in the exosomes. And then the same with fatty acid synthase and ARs, we saw increasing tumor growth and increasing cancer cell growth. So exosomes were isolated. We saw that at the protein level, they had the ability to produce or invoke tumorigenesis, and then we also saw at the mRNA level that they were able to do the same. Once we did this, we looked at the MCF10A exosome treatment. So we treated the non-cancerous cells, those are the MCF10A cells, with MCF7 exosomes. Those are the cancerous exosomes. And we did a proteome profiler, which is kind of similar to a Western blot, looking at specific antibody arrays containing about 84 proteins associated with tumorigenesis. And this is the first result of the proteome profiler. This is just an image of the membranes. So this is for a visual. Here are the control. So at the control, you see that there was no exosome treatment on the MCF10A non-cancerous cells. And here's an example of IL-8, and this IL-8 is normally seen in HER2 positive cancer cells, such as MCF7. And you can barely see it here. But then after exosome treatment of the cancerous exosomes, you can visually see this expression of IL-8. And so this is just showing you visually what this proteome profiler is showing us. So after that, we're looking at the average fold expression. So we had a 6.9-fold upregulation of proteins and then an average of 10-fold downregulation between only 3 proteins. So of those that we went through, only 3 had downregulation. And this is showing the fold expression change and then the proteins. The first protein that had a downregulation, meaning that when the exosomes, cancerous exosomes were treated onto the non-cancerous cells, it was downregulated, not upregulated, is prolactin. And that would make sense because prolactin is actually seen for like milk secretion, so it has nothing to do with cancer. And in this case, it kind of correlates, we didn't test this, but it makes sense that also sometimes it's seen that women who breastfeed have a decreased chance at developing breast cancer. And then the second one that was downregulated was HIF1 alpha. In this case, we would expect it to be upregulated. It's normally seen in cancerous cells. So with that, we'll have to go back and probably try this again and overexpress it and see if that was just an error there. But then upregulated, we see the progesterone receptor. And the progesterone receptor is normally seen positive in any kind of cancer. So as we see MCF7 cells that we were looking at, the exosomes of these, they contain a progesterone receptor. Increased progesterone normally indicates increased cancer cell growth. And then finally, IL-8, as mentioned before, which is seen in HER2 positive cancers, such as MCF7. So we applied MCF7 cancerous exosomes on non-cancerous cells and saw that there were an upregulation of these cancerous proteins within them. So with that, we were able to say that exosomes released from cancerous MCF7 cells affect the genotype of the recipient non-cancerous MCF10A cells by upregulating cancer-inducing proteins. And so this is just a cancerous cell in the breast. This is showing exosomes can be released from this, causing a field cancerized cell, which eventually could turn into a cancerous cell. Moving forward, we want to confirm the upregulation of proteins in the MCF10A cells using specific antibodies and Western blotting, as well as functional overexpression of those specific proteins. And then finally, we want to confirm this data with human breast tissue, breast cancer tissues, meaning that we want to confirm before we go on that this is going to be the same in actual human breast tissue, because we were just using those models. I want to thank Mark Bisofi for his guidance and assistance working with me on this project for the last about year almost, and then Sonika Pandit and Dee Mayan for their assistance using the DLS machine, and Chapman University Schmid College for funding and support. Thank you.
Who has questions? Yes. What structures are actually in an exosome?
And an exosome, so it's an extracellular vesicle. It's the Sorry. So it's an extracellular vesicle. We don't know a whole lot as of right now about specific things about the exosomes. We're looking just more at the fact that we know that they can affect cell function and behavior. And that's why we were able to see from previous literature, like in the prostate cancer, that they would probably upregulate cancer and induce field cancerization. But I don't know specifically for the exosomes, their makeup. All I know really is just that they're extracellular vesicles.
Is there a way to stop exosomes from spreading, or would you just have to like cut more out, like when thinking of the exosomes to get rid of those too?
So yeah, that's what we're kind of curious about too. For right now, we're just looking at, we want to confirm that it is possibly the exosomes causing this secondary tumorigenesis. So once we confirm that, then we can kind of take a step back and look at how to go and treat that, or how to make sure. As we know right now, the reason is like when we go into surgery and get a lumpectomy, they remove what the surgeon thinks is all of the cancer. And they can see, okay, I've gotten all the margins. However, we know that there's probably something they can't detect with a scaffold, you know, and something else that they'll have to go back in and look at more of the biochemical level. So eventually, hopefully, we'll get to that point once we can confirm that you know, that this is what is happening.
Yes.
Do you think any of like, there'd be like a way to like do a screening of exosomes?
That's a good question. I want to know. I don't know. I know.
Really cool.
That would be cool. I think that possibly, I don't know, right now it would just be our own thought. We see an upregulation and say, oh, okay, it's maybe the exosomes. Like maybe if you screen and see an upregulation of IL-8 and you would assume, oh, maybe the exosomes are doing that. But right now I'm not quite sure how to screen.
I mean, what's the sort of non-pathogenic function of exosomes in general? What are they, yeah?
Yeah, so normally I think that, because they're seen in all of our body, I think that they would help with like cell function behavior, possibly in positive ways as well, by upregulating certain proteins, maybe in the case of like, I don't know, trying to think specifically of an example, but anywhere else in our body doing things where you would upregulate proteins or downregulate in a sense too.
One last question. I'll ask a question. Excuse my ignorance on this topic. I'm not a biologist, but is the cancer that's induced by, if these induced cancer on the cells, would that be a different type of cancer with its own mutations to it?
I am not exactly sure about that. So what they're doing is they're just kind of inducing this field. So they kind of prime the tissue for turning or becoming cancerous. So I would assume that it's probably, they're coming from the cancerous cell. So they contain what that previous cancer cell had. So I would think that they would have the same qualities as that, which they were coming from, which they were being excreted from, the cancer they were being excreted from.
And the last question is, if you wouldn't mind sharing with us what your future plans are?
Yes, so I am technically class of 2023, but I'm graduating at fall, so I am still finishing off one last semester at Chapman, but I'm applying to optometry school this summer. So that's my hope of where to be. So, but yeah, so thank you.
All right, so for our next presentation, we have Zeri. Gary was in my general chemistry course 4 years ago. And when we started talking about quantum mechanics, this was something that excited him. So he kept coming to my office and just asking questions, which I thought was great. And he's a computer science major. And so then he asked if he could start taking code to actually do some quantum mechanics. And I said, yeah, sure, why not? It sounds like fun. And this has just kind of ballooned into a program of its whole life, of its own life, that Gary has been working on for the last 20 years. We're talking about that.
So, hi everyone, my name is Gary, and today I'll be talking about my research I've been doing for the past 4 years in a pedagogical computational chemistry package for Jupyter Lab. So then, to begin with, why even do this? What's the point of this? And before I answer that, let's start off with what is computational chemistry? To put it simply, computational chemistry is a combination of computer science, mathematics, and chemistry theory to create a computer application that simulates the outcome of the reaction. Now, the reason as to why this is useful is that it's cheaper and faster than actually doing the reactions physically. For example, if you want to discover some new material, and let's say you have, I don't know, 1,000 reactions you want to do for 1,000 materials, for what's going to be the best thing for what I want, right? You can invest all that money and time in doing the physical reactions, or you can run it virtually on a machine. And the machine won't tell you, this is going to be the perfect, this is what you're looking for. It's kind of going to be like, these are the experiments that did really well. These are the ones that did okay. These are the ones that just completely failed for what you wanted. And from there, you kind of say, I'll just take the best 10%. So we can cut down, let's say, 1,000 experiments now to 100 experiments. So that's really where the power of computational chemistry comes in. So it's very useful and powerful, but as I said, because you have this combination of these computer science, mathematics, and chemistry, each theory itself is its own field with all its own nuances and difficulties. I mean, combining them together is not an easy feat. So that's kind of why I wanted to create this package where it's like, you can kind of start off saying, okay, this is the theory in the textbook, which in the lab we can represent as latex. From this latex, then we can actually now start writing Python code and actually showing, okay, step by step, this is we're going from math, code, and really just understanding what is, because that in and of itself, its own process, unique, like how do you take the math and make it into code? So that's kind of the idea of how we illustrate that. Okay, so this is the actual package that I designed and kind of how it's structured. So it's kind of like a birthday cake, if you want to think of it like that, where you have these three being the base of the entire code system: global imports, graphable, and database. Global imports is a simple way for including functions that are things that everyone needs to use no matter what. For example, you need to do integration, you'll do derivatives, you need Planck's constant, numerical variables. That's all in here. Graphable now is that often when you write these simulations, right, you want to be able to graph things out. But the problem that comes up is that each and every time you build something new, you need to write the graphing code over and over. And after a while, that gets a bit tedious. So this is one code that will graph anything built in this package. So no need to rewrite code. Database is because a lot of these computations, often they can take hours or days, and you don't want to have to keep running this code over and over or saving data in text files, which it'll get messy very quickly. So there's a database that structures all the data in. So if you run anything, you can easily save it, load it back up, all built in nicely. From there, we get into kind of the middle part of the cake, where this is more of the computational chemistry theory. I kind of abstracted it out into a sort of a logical units that the computer kind of knows this is how these objects interact with each other. And all these objects, of course, will pull from the three bases at the top. And these right here, generally, any computation will generally start with a basis set made of basis functions and atomic potential, which you'll eventually fit it. Since that's discrete, you want to make it continuous, which happens right here. That's then fit into your operators, which will solve the Schrodinger equation, the fundamental equation of quantum mechanics and also computational chemistry. And then you get a wave function, which you can then feed into other computation, for example, Frank-Condon computations. But right, you have this data constantly moving around. So even if you have these logical separations, you have to usually manually write code that'll move data along. Package does it all for you, right? So you just really, the idea, right, is that you just have to worry, what am I doing? How am I going to do it? And really leave the details of how do I get A to talk to B or B to talk to C to the package. So you just worry about the pure theory and implementing it. And this level right here is actually what the user sees. So they actually work with a harmonic oscillator basis function, a Morse oscillator basis function, an RKR diatomic potential, and they're able to actually build more items of these types under each logical structure. And they'll all fall into this package operating under the same logical assumptions. So this project was built over 2 phases. So for the first year, we had the initial first methods. It started off with Hartree-Fock, and then the goal was to actually create harmonic vibrational functions, to be able to compute those out. And when we're doing those computations, the goal was just LaTeX and kind of let's illustrate what's really happening here. But what I noticed was happening a lot was that I ended up writing a lot of duct tape code all the time. So for example, I built Hartree-Fock, okay, wonderful, now let's graph it, duct tape code to graph it out. Okay, let's add in the harmonic oscillator, but now these things need to actually talk and go into the Schrodinger equation to be solved. So duct tape code everywhere. And every time I added something new, I'd have to kind of destroy what I built and rebuild that infrastructure. So eventually I thought, why not just build one static infrastructure that's always the same, and I'm always operating under these assumptions, which is kind of where that structure came from, or just naturally arose from this, these data management issues. Either I can keep dealing them as they pop up, or just one solution, and I can really the pedagogical purposes of this. So that's kind of how this came up. So then once we had that structure in place, I could really just go and focus more on just the theory and implementing new methods and new simulations. So other ones I've been doing potential energy search methods. What these are is that this is for diatomics, right? So diatomics are just 2 atom molecules. And as the molecules get closer and further, they feel varying amounts of attraction or repulsive forces. This potential energy surface methods computes out that, and it is that right there. So that kind of represents how attractive or repulsive they are as you get closer or further together. Of these, I've had 3 that are working in the code. We have Hartree-Fock, which is the first I put in, RKR. And you can also import data from any other source. For example, if you have DFT, you can just load up that file and it'll run in the program under the same assumptions as well. From there, we now have the energy surface fitness, because the potential energy surface methods are discrete methods. You just get a plot of points. You can make them continuous technically, but they're very computationally intensive to the point that it's just not feasible from a time perspective. Just use a calculator, let's say 20, 30 points, and then fit them with a continuous function that looks like that curve right there. So initially that starts out with just a bunch of points, and those points are then fitted with a function. And of these, we have 3: Morse potential, extended Rydberg, and multiple Morse potentials. Extended Rydberg is the plot right there. Multiple Morse potentials of this is the most accurate. And that's involving you just fit 3 points for one potential, and then you kind of add in a second one, and that way it just really fits the points closely. From there, we have our basis sets. We have harmonic oscillator and Morse wave functions. Of these two, harmonic oscillator turned out being the most accurate. And then there are also other tools just to help out. The main one being a NIST web scraper, where NIST is a website where spectroscopic data is stored online. This kind of lets you just immediately download it and not have to go and copy paste values in. And this is then generally fed into RKR, as this one analytically will compute out this curve, as opposed from acting from an ab initio standpoint, meaning that let's think about all the electrons and all the protons and all the possible ways it can interact to determine what's really happening. This kind of just says we kind of have the experimental values, let's just build it from there. In terms of other features, there are 3 main features. One is multiprocessing, as this can get pretty slow because you're doing all these intervals and there's a lot of stuff going under the hood. Multiprocessing lets you spread across multiple processors, and depending on how many cores you're splitting across, you'll end up with 50 to 80% speed improvement. That's what I've seen. It's also optimized for online binder access, which is a website, so you don't actually have to download this code to use it. You can go online to a website, there's a link, and then you can just use it there. It's a lot slower than running it on your own machine, but it saves you the time having to download and install all the necessary packages. From there, I don't know how many of you actually work with plotting in the sciences, but normally you would use something called matplotlib, which is a bit old-fashioned. This is Plotly, and because it's a newer package, it gives you interactivity, and I just think generally looks a lot nicer in comparison to the standard graphing package. Okay, so in terms of applications where this has been used, there's been 3 main uses. Dr. LaRue has used this in his own research, where I computed out the Frank Condon factors, and then he's compared this to spectroscopic data that he's had to kind of figure out, is there any sort of vibrational dependency in this data? He eventually came to the conclusion that there was none, so it was useful for his research in that sense. From there, it's also used as a pedagogical tool in his physical chemistry lab. And actually, the lab is right there. The purpose of this lab really is just kind of focus, what's the difference between an anharmonic versus harmonic potential? And this code right here, this graph, lets you interact with that idea and those objects completely without writing any code. You can kind of just mess around with the settings at the bottom and really just get an intuitive feeling for what's going on here, as well as also quantitative since you have the values right there. And of the 8 students that took a survey on this, 100% of them say that this was a significant improvement over the old way of learning the material and that they felt they learned more. Now, I understand that's a small sample size, definitely not indicative or statistically significant, but it's still very exciting that of the 8 people that did use 100% were like, this is a lot better than what we were doing before. And then Dr. Lou perhaps uses a molecular spectroscopy course for a slightly different purpose. In the other course, it was used at a high level, meaning that people just kind of use it to look at the graph and play around with these settings and kind of get an intuitive feeling. For this spectroscopy class, he plans on using it to actually understand what's actually happening under the hood for the Frank Condon computation in terms of nuts and bolts. How is this code actually functioning at that level? So that'll be exciting to see. And to conclude, this was kind of, I had so many more ideas for what I wanted to do with this. And unfortunately, there just wasn't a time to actually implement them. This is kind of other ideas I wanted to take this in. Hopefully, maybe in the future, someone else can pick it up. It is fully open source, so that's a good part of it. One thing is that it's very slow if you want to do really intense computation. So I was thinking, right now, everything's running on your CPU, but if you could move the integrals, which tend to be the slowest part, onto a GPU, which is optimized for running multiple things simultaneously in parallel, I believe we would see significant speed improvements. On top of that, so many more methods I want to do time dependency, molecular dynamics, DFT, but just not enough time for it. And then also, of course, expansion to 3D as opposed to just standard 2D diatomic systems. And in terms of the pedagogy of it, right, this was only from one lab that was we only tested this in one lab at Chapman with about 8 students who replied to the survey. I believe that in order to really get a good feel of how effective is this, we need to test it with more students at Chapman and also students outside of Chapman. And even if we don't get exciting results, 100% everyone loves it, I still think that's important that we see how can we really improve this to make it the best that it can be. Like what's good, what's bad, what's working, what isn't. And to do that, we definitely need a lot more data on that. Oh, in terms of future projects, I plan on, as at the end of the semester, I'll be graduating, I won't be working on this anymore. Other projects I'll be picking up is a LibreText simulations. So LibreText is an online website where there are free textbooks for chemistry, physics, sciences, mathematics. And I'm working with Dr. LaRue to develop online simulations, similar to PhET simulations, if you've heard of those, which is to kind of move their mouse around, play around with, for example, a steam engine or electrons in a box to really understand how do these concepts apply to more visual level, like intuitively what's really happening here, as opposed to just seeing all this theory and mathematics on a white piece of paper. And another project I'm working on with Morgan Grimes is actually going to be developing a VR headset application where you can actually visualize reactions and what's happening. So you put on the headset and then you can look around and see the reaction happening around you in real time. That'll be kind of interesting to see. And then lastly, thank you so much everyone for your time. And of course to Dr. LaRue for all your help and support. I do really appreciate all the time invested in this. And questions, anyone? Questions?
Maybe could you expand a little bit, because we had talked about a lot of ideas for a
Yes.
future, more than just what's there, for
Yes,
example, using, could you mention some more ideas that we had of what you could do, down the road?
Yes. Oh, in terms of that, I mean, I think the biggest thing if I do continue in this pathway would actually be building, I think moving slightly away from pedagogy and moving more towards practical applications and building a full-on 3D application that runs on your machine and is actually capable of telling, for example, if you're going to test out a drug in a biological system, what will happen in that sense. Which of course that's a lot more big scale and complicated, but that's definitely one pathway I'd like to look into. Also possibly for a PhD, but I'm still trying to figure all that out. Yes.
What are you doing next year? You've got amazing ideas. What are you doing after you graduate?
I'm continuing my master's here at Chapman, computational data sciences.
Awesome.
Yes.
Gary, what made you start on the first model that you implemented? Why was that the one that you chose?
Okay, so that's actually a bit of a long story. I'll try to tell. So in high school, I had an opportunity. Well, I always wanted to be a programmer. That's something I always knew. So in high school, I had a teacher, chemistry teacher, say that they're doing an opportunity for computational chemistry. You can write a lot of code for UCI if you want to in one of their research labs. So I joined that. And I mean, the code was something that I was very excited about, but they also introduced a lot more to like the math and chemistry side of things, which were very novel at the time. So I think it was just like I went there and was like, what is all this magical nonsense, quantum mechanics? So it's just really out of morbid curiosity, if you will. And then I came here, I was, as you said, I had so many questions. And then that's where the first thing evolved from, really just out of curiosity.
Gary also ended up because of this, he ended up also pursuing a chemistry minor.
Yes, that's definitely 100% true. Yes.
Any last questions? All right, let's thank Gary again. Let's go ahead and get started. So our next presenter is Aviv Zohman. Aviv came to me last year with his own research idea, which is fantastic. He was highly motivated and just ready to get going from the start to start working on this. So he has pretty much come up with this mostly all by himself. My role in this was really just to bounce ideas off of and make sure he didn't blow anything up in my lab. The rest was really all Aviv. So.
Hello, everybody. My name is Aviv Zohman, and for these past couple semesters, I've been working with Dr. Jerry LaRue in designing and building a microwave plasma reactor for graphene synthesis. So this title raises about 4 questions. What's graphene? What's a microwave plasma reactor? What's their connection? And who cares? So I hope throughout this presentation, I'll be answering those questions. So I'll first start off by describing what graphene is. And in doing that, it's helpful to talk about graphite, the form of carbon that we're all familiar with that's commonly known as pencil lead. The molecular structure of graphite resembles a deck of cards, where each card in this deck is a sheet of carbon atoms arranged in a honeycomb lattice. And if you remove one of these cards from the graphite deck, then you get graphene. So in other words, graphene is just a single layer of graphite. So the natural next question is, so what? I mean, who cares about what graphene is? Well, it's graphene's extreme thinness. It's only one atom thin, and molecular structure that give rise to a unique combination of incredible properties. For example, it's 200 times stronger than steel. It's the strongest material ever measured, yet it's also the thinnest. It's more electrically conductive than copper, more thermally conductive than diamond. It's optically transparent, yet so dense that it's impermeable to all gases. And on top of all of that, it's more elastic than rubber. And no one material has ever encompassed all of these properties before, so graphene has been coined as our era's ultimate material. So graphene will revolutionize every industry imaginable and certainly create new ones. We'll get lighter, stronger structures in the field of architecture, lighter, safer, stronger vehicles in the automobile and aeronautical industry, and we'll see, the electronics sector revolutionized by transparent bendable electronics, and we'll see energy harvesting devices like solar cells and batteries taken to the next level. But we aren't yet reaping the benefits of graphene because it's so hard to produce. Most processes today produce graphene that's impure, flaky, wrinkled, cracked, and multi-layered, and all of these diminish graphene's properties. It's only after determining a method that will consistently produce pure single-layer graphene that will really be propelled into this graphene age. And this is the aim of my research. So microwave plasma chemical vapor deposition is a promising method to solve this graphene production issue. But this title is formidable, so I'll break it down one at a time. Deposition is just a phase transition when a vapor transitions to a solid. Chemical vapor is a chemical that readily vaporizes, and a chemical implies that it's from the gas phase, not solid or liquid phase. The plasma is the fourth state of matter, and in describing a plasma, it's helpful to talk about the previous three states. So in a solid, the particles are closely packed together, in a liquid, a little further apart, and a gas, they're free-flowing, take up the volume of their container. In a plasma, the energy of the system is so high that these particles are actually stripped of their electrons, and we are left with these negatively charged electrons and positively charged ions that are continuously colliding into each other, creating even more charged particles. And so in essence, a plasma is a soup of charged particles, free radicals, excited state molecules, and most notably photons, which give plasma its characteristic glow. An example of plasma that we all know is lightning. In this case, it's the air molecules being ionized. A microwave plasma just means that this plasma is being ignited by microwave radiation. It's specifically 2.45 gigahertz here because that just so happens to be the frequency of microwave radiation in every household microwave oven. And microwave plasma is essential to this process because it's the source of activation energy for compound formation at the substrate. So to bring it full circle, microwave plasma chemical vapor deposition utilizes microwaves to generate a plasma, which then deposits its contents onto its desired substrate. It's a method to produce materials from the bottom up. A microwave plasma reactor is the module in which microwave plasma chemical vapor deposition occurs in. And it's this very system that I spent the past few months designing and building with Dr. LaRue to form graphene. So I'll first walk through its components and then the actual system afterwards. So first, the vacuum pump. This is to get rid of any contaminants in the system because we want a pure graphene sample. Additionally, this low pressure environment actually allows the microwave radiation to ionize and dissociate the molecules, and this is why you don't generate a plasma every time you turn on your microwave at home. Microwave radiation is simply too weak to affect all of those particles in ambient conditions. Next are the gases. Argon isn't used during deposition, but prior. It's used to clean out the system of any contaminants and therefore decreases the system pressure significantly. Next is methane. Methane is the primary feed gas. That is, it's the gas that supplies the carbon for graphene formation. It has a low dissociation bond energy, which is advantageous for forming these reactive carbon species, as shown in this last line here, which enable the graphene growth. Hydrogen also plays a critical role at the actual substrate, where it also catalyzes carbon activation. In this case, it works by dehydrogenation of methane, as shown in this first reaction. And it's only once we have this reactive carbon species that we get graphene, as illustrated in the second reaction. And both methane and hydrogen are fed into the system by flow meters because their ratio is critical to pure graphene production. So next is the nickel sheet. This is our substrate. This is where the graphene actually forms. Nickel is chosen because it has a high carbon solubility, which enhances graphene growth, as well as promotes crystallinity of the graphene film. So if we take a closer look, we see that those reactive carbon species that I mentioned earlier are bonding with this nickel surface and forming graphene. And then this whole reaction takes place in a vacuum chamber that ought to be made of quartz, because quartz is strong and can withstand high temperature swings, which is necessary to withstand a plasma for a long time. Additionally, it allows microwaves to pass through it. And lastly, everything is enclosed in a household microwave oven because these are inexpensive, easily obtainable, and provide the microwave radiation necessary to ignite a plasma. So enough of the abstraction. Here are some concrete images of the system. Here is that vacuum pump that I mentioned, and this giant exhaust line is the exhaust line that feeds into the building's exhaust system, just getting rid of any byproducts of the reaction we don't want. And then this guy, there's a lot going on in these pictures, but this guy here is the vacuum display, which surprisingly displays the pressure of the vacuum chamber, which is important to know. And that connects to this vacuum gauge, which is in line here to this vacuum inlet. And that vacuum inlet channels into this aluminum block here, which is the base of the deposition chamber. It has two tunnels, one for this vacuum inlet here, and then the other for the gases that enter through this shower head orientation. And the reason it's a shower head is because we want the molecules accelerating directly down into the substrate, which would lie about here. This is a Viton rubber gasket, which interfaces this aluminum block and what should be a quartz container. This one's been replaced. And then here is this gas line, and how gases enter through this system is by the following configuration. As you can see, there are at least a couple valves between every gas between their tank and entering the system. This is it's a safety precaution. You have to really want a gas to be in there. You have to know what you're doing. And additionally, it avoids compromising the vacuum. And then, as you can see, the hydrogen isn't actually connected to anything yet. That's because I've only just finished setting up this system, and I'm taking it one gas at a time still. And then here's that methane tank, which is flowed through this, or fed into the system by this flow meter, which I've balanced by jerry rigging on this platform. And then lastly, here's this argon tank, which actually has two inputs to the system. One, if you follow this line, actually populates the entire microwave oven with argon, because in the low probability event that methane seeps out of the chamber, we don't want it reacting with oxygen at high temperatures. And here's that argon line that flows directly into the system, as we've seen previously. And so, to put an even better face to a name, I thought it'd be fun to actually watch a deposition process to see what it looks like. I'll leave it unnarrated, so feast your eyes. Oh. Okay. Yeah, I know. Okay, so unfortunately we can't see the video right now, but you can imagine this beautifully purple plasma just shocking your eyes. So fortunately we can't see that. After doing this deposition, I characterized these results using a scanning electron microscope. And so what we can see here is a comparison between the bare nickel substrate, just what the nickel substrate looks like under this microscope, and a couple deposition samples. They're virtually identical because the production parameters are still young, so this is unsurprising. Additionally, this leads me into the future directions of this project. I'd firstly like to use Raman spectroscopy to characterize the results, because Raman is the ideal graphene analysis tool. It really tells you how many layers your sample is and how impure it is. Another future direction is using induction heating to increase plasma density. And by plasma density, I mean the quantity of these reactive carbon species for graphene formation. So how this would work is I'd wrap the chamber in a copper coil, send an alternating current through it, and what this alternating current would do is it produces an alternating magnetic field, which would induce a voltage in the plasma, which I hypothesize will increase the energy of these electrons and charged particles in there, creating more successful collisions, which is important for graphene growth. And this isn't, theoretical. I've actually already set this up. It's just a matter of carrying out. Here is that copper coil wrapped around the chamber, and it's connected to a water coolant system here that leads into this bucket of water with a water pump, and then here's the electrical circuitry that provides the alternating current to it. Additionally, I like to confine the plasma with magnetism. See, plasma has a really interesting dynamic with magnetism, as illustrated in this first picture, where the magnetic field line spiralizes the electrons and ions. So taking advantage of this behavior, I'd like to wrap the chamber in permanent magnets that are alternating in magnetic orientation so that their field lines feed into one another. And then you can imagine that these charged particles are spiralizing in a localized area. And this is to keep the system, the heat of the system in a specific area, because I don't want the substrate getting so hot. And this leads me to my next point. I'd like to install a thermocouple in the chamber to measure the reaction chamber, because the ultimate aim is to deposit graphene on insulating substrates like plastic and glass, which would melt at high temperatures. And then lastly, I'd like to use machine learning techniques to determine the optimal production parameters for graphene growth. And this is pretty straightforward after I set up Raman spectroscopy and run a few trials and have enough data to do this. And while I'm very happy with the current state of this project and look forward to the forthcoming work, I owe a lot to the Center of Undergraduate Excellence for helping me kickstart this project, Mayuki Weldon and Matthew Snyder at Fowler's Makerspace for helping me machine parts, my dad, Aviv Zohman, for helping me machine parts, inspiration to take on this project from Julie Schultz and Robert Murray Smith, Dr. Islam for helping me characterize these results in such short notice, and Dean Lyon for referring me to Dr. LaRue, who I owe a very special thanks to for allowing me to establish this independent project in his lab, despite its unrelatedness completely to his research agenda, and then giving me the freedom to pursue it limitlessly, except for safety, of course. And with that, I'd like to thank you all very much for your attention.
Last questions.
Okay, this is because I'm really ignorant around this. So how is it that you just get one graphene layer and you don't get one on top of another?
Right, that's a good question, and I can't say with confidence the specific physical chemistry of that process, but hydrogen also plays that I didn't mention. Hydrogen plays a critical role in acting as an etchant, and so it creates a sort of self-limiting nature of the graphene growth. But I don't really want to extrapolate any further because I don't want to provide a hand-wavy explanation.
Other questions? Yes.
So how important is it that the microwave field, which is obviously the key energy source of making all this happen, how important is it that that's fairly across the space? And then part of the reason I asked the question, being an old RF sputtering guy from years ago, we know that commercial microwave ovens tend to be rather uneven in terms of their heating, and that's why there's carousels and turning food around and all that sort of thing. So is uniformity important, and would you consider moving to a different microwave source for future experiments?
That's a good question. I'd imagine that uniformity is very important. This is why I'll just bring it back up. This is why I plan on using induction heating, to increase that sort of uniformity, I guess, the energy of the system. Additionally, I'm using a microwave oven because it's just convenient. It cost me $20 on OfferUp. Yeah, it can't beat that. I have investigated actually rewiring the microwave, like using a transformer, but that would, I wouldn't risk messing with lethal components like that. So this is a good technique. But yes, uniformity in terms of the reaction is certainly important. Yeah.
Stephanie?
Oh, what are some safety precautions that you take to make sure you don't blow up the lab?
Yeah. Right, so the very first safety precaution is, having a very tight vacuum. So this gets down to, about 100 millitorr, and we live in 760 torr region. So that means that the container itself can't just accidentally fall over, when there's methane inside of it. Like it's really stuck in place by the vacuum created. Additionally, we use argon to, we flood the entire microwave oven with argon in the case that methane would seep out and react with oxygen and at high temperatures to prevent that. So the methane would just seep out and bounce into argon molecules. So that's a preventative measure. If you'd like to, offer any more because you're worried about your own safety, I understand. So just let me know.
There's also plenty of shut-off valves around the whole for gas wells and stuff. Yes.
How did you determine that quartz was the best source for the timber?
That's a good question. So the best answer is trial and error. So I went through a couple different phases of trying out different vacuum chambers. Initially just with glass, regular glass, I think it was a Papers jar from Trader Joe's, and that cracks. Unfortunately, none of these resulted in implosions, but it cracked, so I knew that wasn't good. Additionally, I did research into this, so it wasn't just swimming for the fences. And then I went to borosilicate glass, which is an even stronger type of glass, but I made a mistake there because borosilicate has a high tensile strength, but it can't really withstand temperature swings very well. So that's when I realized that I need quartz. That it was all about temperature, not strength, really.
Time for one more question. All right, well then, I'll ask you, if you mind sharing with us, what are your future plans?
So I'll be getting my master's degree in material science and engineering at Columbia University next fall, where I aim to advance this graphene research.
All right, thank you. All right, so let's get started. It's my pleasure to introduce Chelsey Cortes. She has been doing research since about her freshman year. She started as shadow student in my group. Now, for the last 2 years, she's been really just kind of one of the driving forces behind the project, just looking it forward, one of the lead researchers on the project. She is also the first of the generation of Orange High School students to graduate from Chapman. So she was amongst the first of the Simon Scholar program. So Chelsey, it's all yours.
Hi, I'm Chelsey. So I've been working in the research that I've been doing on synthesizing bimetallic core shell nanoparticles that have the potential to work as a novel type of photocatalyst. So my presentation. So catalysts are vital to the petroleum industry. And so you might be wondering, why do we need these photocatalysts? Well, we need photocatalysts because currently with the problems that we have, specifically global warming, we need to decrease pollution emissions, specifically hydrocarbon emissions and carbon monoxide pollution. And two of the biggest contributors to hydrocarbon emissions and carbon monoxide pollution is the petroleum industry, as well as automobile exhaust. And so some ways that we can potentially mitigate these emissions are through hydrogenation and CO oxidation reactions. So the CO oxidation reaction is the one on top. And then the hydrogenation reaction is right here. So we can either convert carbon monoxide into carbon dioxide or into methanol. But for these reactions specifically, it requires catalysts, and sometimes catalysts that are being used are not very efficient, and sometimes they can be expensive. Sometimes they use some of the catalysts that are being used are monocatalysts, such as silver and gold, that by themselves they do work, but they don't provide enough energy for charge transfer for specific chemical reactions to move forward. So what we propose is to synthesize bimetallic nanoparticles. This is an example of kind of what they kind of look like. So the bimetallic nanoparticle would be made by coating the titanium nitride nanoparticle with a transition metal shell, and this would form a green solar to chemical photocatalyst. I'm also a biochemistry major, so some of the research I've been doing also is to look at how these bimetallic nanoparticles can also work in like the biochemistry area. So what I found really interesting was that ruthenium gold nanoparticles, which are bimetallic nanoparticles, can also bind to DNA and can be used as luminescent probes for various biological applications, but that would be for further research. I'll be focusing on this for this presentation. So titanium nitride is the core that we are using, so the inside of the nanoparticle. So the titanium nitride, we specifically chose this because it has localized surface plasmon resonance, and localized plasmon resonances, kind of see here, are a density of electrons. You can kind of think of them as a sea of oscillating electrons that are confined to the metal catalyst, so in this case, titanium nitride. And when these localized plasmon resonances are hit with light, they become very energetic and then they decay. And then these result in highly excited electrons, or what we like to call hot electrons, that can then promote excited state chemistry. They specifically can, the electrons can move into the antibonding orbital and promote reactant surface bonding. There we go. And the result is photochemistry. So here is an example. I've been talking about the bimetallic nanoparticles. So this is kind of what it looks like. So we have the titanium nitride core, and then we have a chemically active metal shell. So currently I have been working on creating, two types of bimetallic nanoparticles. So one with a ruthenium transition metal shell and one with a rhodium transition metal shell, around a 20 nanometer nanoparticle. And then we also have rhenium for future work. So we are currently working on creating a synthesis procedure for this specific bimetallic nanoparticle. So, what we're trying to do with the transition metal shell is either ruthenium or rhodium is to modify the reactant surface of the titanium nitride nanoparticle, which will allow for selective bond activation of certain reactions. So specifically by putting the ruthenium rhodium shell, which has a 4D, which are 4D transition metals, we can selectively activate in our hydrocarbon oxidation reactions. We can selectively activate oxygens, which can then form carbon dioxide. And this, so when we do this, by adding the transition metal to the titanium nitride core, by themselves, the plasmonic metal core does not work, is not super efficient. And this is because its anti-bonding position is kind of far from the Fermi level. So by putting the transition metal core around the plasmonic core, the transition metal shell around the plasmonic core, we bring the anti-bonding states closer to the Fermi level. The Fermi level, you can think of it as the energy required to move an electron, to excite the electron into an anti-bonding orbital. So the electrons will jump into the anti-bonding orbital reactant surface bond of oxygen again, and then promote the excited state chemistry. So for the ruthenium, which is the first bimetallic nanoparticle I will synthesize, there was two methods. So the first method is the sodium citrate method. So we used ruthenium 3-chloride and sodium citrate in nanopure water. And we made the solution, and then we boiled it for 5 minutes in a reflux reaction, kind of like here. So we have the our solution here, and then we boil it for 5 minutes, and then once done, we let it cool, and then we centrifuge our solution so we can obtain just the nanoparticles, and we get rid of the any sodium citrate that's left over with nanopure water washes. And we also synthesize these at either 1:1 ratio, which is a 1:1 ratio of ruthenium to titanium nitride, or 2:1 ratio of ruthenium to titanium nitride. And the second method for creating a ruthenium titanium nitride bimetallic nanoparticle is the oleolamine solution method. So in this one, we use an oleolamine solution, which is like an oily substance rather than water. And triruthenium dodecarbonyl, which is right here, to create the bimetallic nanoparticles. And they go through the same process. So we solution and we allow, or the reflex reaction. This, however, instead of 5 minutes of boiling, we do 30 minutes. And then once done, we centrifuge and wash away the oleolamine solution with ethanol washes. And these, similar to the ruthenium titanium nitride core, we have different ratios of 1:1, 2:1, and 4:1. So to see how these nanoparticles have been looking, we use various characterization tools. The first one we have been using, and the main one, is scanning electron microscope, and this just kind of gives us information of the surface morphology and just overall structure of the titanium nitride nanoparticles that we have been making. So here's the machine, and you can kind of see what they kind of look like right here. And we also use energy dispersive X-rays, which give us a view of what elements are in our sample, and just confirm that we do indeed have titanium, ruthenium, rhodium, whatever we have been using to synthesize our bimetallic nanoparticles. And then we also use transition electron microscope, which is kind of like scanning electron microscope, just a better version of it. So it just gives us better structural information with a higher resolution of the bimetallic nanoparticles. So this is the characterization results of one of our samples. So this right here on the left is 100 nanometer SPM image of titanium nitride ruthenium nanoparticles at a 2:1 ratio. And these range in a size of 50 to 70 nanometers, which is kind of big. If you remember, I said that our titanium nitride core shell by nanoparticles by themselves are 20 nanometers. So this is kind of on the bigger side. It's specifically 70 nanometers, so we want them to be a little bit smaller. But here we have an EDS image, which confirms that we do indeed have ruthenium in red and titanium in green. And then a little bit of like pink color for the nitride. And then we also have the oleolamine characterization results. So here we have a 100 nanometer SEM image of the titanium nitride ruthenium nanoparticles at a 1:1 ratio. The size is around 40 to 55 nanometers, which was kind of expected because, again, our nanoparticle is about 20 nanometers. So these are already much better results than the other method. And then these are, if compared to the other one, they're a little bit more defined and more spread out, which we liked. And then your EDS image of the ruthenium and titanium nitride. And here it's a little small, but we have 38% of our ruthenium, 36% titanium, and 27% nitride. If we just look at titanium and ruthenium for this specific EDS image, we do have like kind of a 1:1 ratio of our titanium to ruthenium, which we really liked, but this overall is not a whole image of our entire sample. It's just a small take of our sample. So it's not, it doesn't give us the whole image of like, oh, it is 1:1. And so we did run TEM, which gave us even better information about our nanoparticles. And what we found from TEM was that the sodium citrate method does not produce nanoparticles with an even shell distribution of ruthenium, but oleolamine method does. So here is the TEM EDS data on the left depicts even distribution of titanium, here titanium, over the ruthenium. So if we were to overlay these images, we would see a good distribution of ruthenium over the titanium. And then this is not like a single nanoparticle. We see the ruthenium and titanium, it's evenly distributed. Here, however, we do not, this is the This is the sodium citrate method. We do not really see a good distribution of the ruthenium over the titanium, so we concluded that we would go better with with the loylamine method. So rhodium is our second sample that we're using, and so this sample uses 3 different elements and a loylamine solution similar to the other loylamine method. And our rhodium method characterization results were also pretty good. These are 100 nanometer SEM image of titanium nitride rhodium nanoparticles at a 2:1 ratio. They have a size around 50 to 60 nanometers. And these show really good even distribution, which we really liked. This is an image that's kind of zoomed out at 300 nanometers, of the same sample. And that 300 nanometer sample, we took an image and we found that it does have rhodium and titanium nitride, which we really want. And it has a 36% rhodium, 62% titanium, and 2% nitrogen. Again, these are a little bit weird just because this is only a part of the sample, not the entire sample. So for future work, we are going to be testing the photocatalytic activity of our samples just to make sure that they are working as photocatalysts. And we will be doing surface enhanced Raman spectroscopy. To study the vibrational energies that make up the coarse nanoparticles and just better understand the model structure. And we will also observe the reaction as it moves forward. And right here, we'll be using Raman reactor chamber and the mass spectrometer, just to, again, assess the photocatalytic activity and just efficiency of the overall coarse nanoparticles, in addition to also studying rhenium and creating a synthesis method for rhenium. References, and then I just want to give special thanks to Dr. Ru for her mentorship, Dr. Benji Zhang, who's also my mentor, and then Ishan sitting over there for all his help and explaining everything that I do not know. And he's also the one who's building the Raman reactor chamber. So once that's finished, we'll be testing that. And then Dr. Islam for helping me with the SEM and EDS, and Stephanie, who's also sitting over there, for helping me do synthesis, which takes quite a while. And then UCI Material Research Institute, they actually have the TEM tool that we use. We don't actually have it here, so thank you for letting us use that. And then also CUE, SURF, and for help.
I think you can go back to your first SEM analysis on the loyalty methods. With the EDS, with sodium citrate, you see a lot less nickel versus that pitch. What do you think could be the reasoning for that, or the variations?
So you're saying like why is there less nitride?
Yeah, why is there, like, you see it in that oleolamine EDS, less, the titanium versus the nitride?
Oh, okay. I'm not quite sure. I think it might have to do in the reaction. It probably might be going away, but I'm not entirely sure. I would have to get back to you on that.
You spoke earlier on possible DNA interactions. What do you think are possible future routes for that?
So when I was doing the reading, they actually interacts with the nucleus and cytoplasm. So I think that would be interesting to see how perhaps like follow maybe cancerous cells or something like that. I'm not really sure, but also the ones that were being studied was ruthenium and gold. We use ruthenium titanium nitride, so maybe just looking to see if the titanium even reacts with the DNA would probably be interesting.
Yeah, so this actually, bringing up the cells research was piggybacked off of my research, which was on platinum drugs, and I also originally had these molecules for catalytic purposes. And while I was doing my research, I did see the gold come up, so it would be interesting to see if the rubidium has the same effect or if there's a different effect on cancer cells.
As you said, the oleamine method is better than the sodium citric method, and why do you think that overall it had better results and why they were smaller in size for oleamine versus sodium citrate?
Yeah. I'm not sure about that. It probably has to do with the either the oxidation or reduction reactions that happen. Specifically oleamine, like the reduction effect that has on ruthenium. I'm not sure about that. Probably look into that a little bit more.
Sean?
From the piggyback a little off of Stephanie's question, we saw that for the ruthenium shell, you got around 70 nanometers. For the rhodium, you got 40 to 55. Do you have any reasoning why that?
Are you saying, sorry, so for this specifically, ruthenium and just ruthenium, right?
Yeah.
And so sodium citrate versus oleamine. I'm not entirely sure why, but I do know that it's just I think the sodium citrate is just not working as great with the titanium nitride particles. So that's probably why we're getting larger nanoparticles versus oleamine, where we are getting a little bit smaller, more defined nanoparticles. I don't know if that answered your question.
How might the rhodium be different? Is rhodium a better shell metal than
Oh, okay. I'm not entirely sure about that part. It might have to do with their structures. I know that we're using for rhodium. We're using rhodium 3-acetone, which has like a specific structure. I didn't put it up here, but it's a little bit more complicated, and I think it's more defined than the other one. So probably might have to do with this, how the rhodium versus ruthenium look. Yeah, I have to do more research about that. They're tough questions.
Is there a last question? Or one more? All right, so Chelsey, would you like to tell us what your plans are going forward?
So over the summer, I'll be working with a gastroenterologist as a scribe, and then I'll be continuing research here. That's all.
And do you have any plans after summer, or is that?
I'll be applying to PA school, the one at Chapman, and then working on my medical school apps.
Great, awesome. All right, let's thank Chelsey.